Although chloroplasts are larger, Figure 1425a, they are organized on the same principles as mitochondria. Chloroplasts have a highly permeable outer membrane and a much less permeable inner membrane, in which membrane transport proteins are embedded. Together, these membranes and the narrow intermembrane space that separates them form the chloroplast envelope, Figure 1425b. The inner membrane surrounds a large space called the stroma, which is analogous to the mitochondrial matrix and contains many metabolic enzymes. There is, however, an important difference between the organization of mitochondria and that of chloroplasts. The inner membrane of the chloroplast does not contain the electron transport chains. Instead, the light-capturing systems, the electron transport chains, and ATP synthase are all contained in the thylakoid membrane, a third membrane that forms a set of flattened disc-like sacs called the thylakoids, Figure 1425c. These are arranged in stacks, and the space inside each thylakoid is thought to be connected with that of other thylakoids, thereby defining a continuous third internal compartment that is separated from the stroma by the thylakoid membrane. Figure 1426. The structural similarities and differences between mitochondria and chloroplasts are illustrated in Figure 1427. Chloroplasts capture energy from sunlight and use it to fix carbon. As we saw in Chapter 3, the overall equation that summarizes the net result of photosynthesis can be written as follows: light energy plus CO2 plus H2O to sugars plus O2 plus heat energy. Although the equation is quite simple, the reactions that allow the process to occur are fairly elaborate. Generally speaking, however, the many reactions that make up photosynthesis in plants take place in two stages. See Figure 3.8. 1. In the first stage, which is dependent on light, energy from sunlight is captured and stored transiently in the high-energy bonds of ATP and the activated carrier molecule NADPH. These energy-producing photosynthetic electron transfer reactions, also called the light reactions, occur entirely within the thylakoid membrane of the chloroplast. In this series of reactions, energy derived from sunlight energizes an electron in the green organic pigment chlorophyll, enabling the electron to move along an electron transport chain in the thylakoid membrane in much the same way that an electron moves along the respiratory chain in mitochondria. The electron that chlorophyll donates to the electron transport chain is ultimately replaced by an electron extracted from water. This electron shuffle splits a molecule of water, H2O, producing O2 as a byproduct. During the electron transport process, H+ is pumped across the thylakoid membrane, and the resulting electrochemical proton gradient drives the synthesis of ATP in the stroma. As the final step in this series of reactions, high-energy electrons are loaded together with H+ onto NADP+, converting it to NADPH, Figure 1428. 2. In the second light-independent stage of photosynthesis, the ATP and the NADPH produced by the photosynthetic electron transfer reactions serve as a source of energy and reducing power, respectively, to drive the manufacture of sugars from CO2. See Figure 1428. These carbon fixation reactions, also called the dark reactions, begin in the chloroplast stroma and continue in the plant cell cytosol. They produce sucrose and many other organic molecules in the leaves of the plant. The sucrose is exported to other tissues as a source of both organic molecules and energy for growth. Thus, the formation of ATP, NADPH, and O2, which requires light energy directly, and the conversion of CO2 to carbohydrate, which requires light energy only indirectly, are separate processes. although elaborate feedback mechanisms interconnect the two sets of reactions. Several of the chloroplast enzymes required for carbon fixation, for example, are inactivated in the dark and reactivated by light-stimulated electron transport processes. Sunlight is absorbed by chlorophyll molecules. Visible light is a form of electromagnetic radiation composed of many different wavelengths, ranging from violet, wavelength 400 nanometers, to deep red, 700 nanometers. But when we consider events at the level of a single molecule, such as the absorption of light by a molecule of chlorophyll, we have to picture light as being composed of discrete packets of energy called photons. Light of different colors is distinguished by photons of different energy, with longer wavelengths corresponding to lower energies. Thus, photons of red light have a lower energy than photons of green light. When sunlight is absorbed by a molecule of chlorophyll, electrons in the molecule interact with photons of light and are raised to a higher energy level. The electrons in the extensive network of alternating single and double bonds in the chlorophyll molecule, figure 1429, absorb red light most strongly, which is why chlorophyll looks green to us. Excited chlorophyll molecules funnel energy into a reaction center. An isolated molecule of chlorophyll is incapable of converting the light it absorbs to a form of energy useful to living systems. It can accomplish this feat only when it is associated with the appropriate proteins and embedded in a membrane. In plant thylakoid membranes and in the membranes of photosynthetic bacteria, the light-absorbing chlorophylls are held in a large multi-protein complexes called photosystems. Each photosystem consists of an antenna complex that captures light energy and a reaction center that enables this light energy to be converted into chemical energy. The antenna portion of a photosystem consists of hundreds of chlorophyll molecules that capture light energy in the form of excited high-energy electrons. These chlorophylls are arranged so that the energy of an excited electron can be passed from one molecule to another, until finally the energy is funneled into two chlorophyll molecules called the special pair. Figure 1430. These two chlorophyll molecules are located in the reaction center, a protein complex that sits adjacent to the antenna complex in the membrane. There, the energy is trapped and used to energize one electron in the special pair of chlorophyll molecules. The reaction center is a transmembrane complex of proteins and organic pigments that lies at the heart of photosynthesis. It is thought to have first evolved more than 3 billion years ago in primitive photosynthetic bacteria. Detailed structural and functional studies have revealed how it functions at an atomic level of detail. Movie 14.6. The reaction center acts as an irreversible trap for an excited electron, because the special pair of chlorophylls are poised to pass the high-energy electron to a precisely positioned neighboring molecule in the same protein complex. By moving the energized electron rapidly away from the chlorophylls, a process known as charge separation, the reaction center transfers this high-energy electron to an environment where it is much more stable. When the chlorophyll molecule in the reaction center loses an electron, it becomes positively charged. It then rapidly regains an electron from an adjacent electron donor to return to its unexcited, uncharged state. Figure 1431a. Then, in slower reactions, the electron donor has its missing electron replaced with an electron removed from water. The high-energy electron that was generated by the excited chlorophyll is then transferred to the electron transport chain. This transfer leaves the reaction center ready to receive the next high-energy electron excited by sunlight. Figure 1431b. Light energy drives the synthesis of both ATP and NADPH. In mitochondria, the electron transport chain functions solely to generate ATP. But in the chloroplast and in free-living photosynthetic organisms such as cyanobacteria, electron transport has an additional role. It also produces the activated carrier molecule NADPH. See Figure 334. NADPH is needed because photosynthesis is ultimately a biosynthetic process. To build organic molecules from CO2, a cell requires a huge input of energy in the form of ATP, and a very large amount of reducing power in the form of NADPH. To produce this NADPH from NADP+, the cell uses energy captured from sunlight to convert the low-energy electrons in water to the high-energy electrons in NADPH. To produce both ATP and NADPH, plant cells and cyanobacteria use 2 photons of light. ATP is made after the first photon is absorbed, NADPH after the second. These photons are absorbed by 2 different photosystems that operate in series. Working together, these photosystems impart to an electron a high enough energy to produce NADPH. Along the way, a proton gradient is generated, allowing ATP to be made. In outline, the process works as follows. The first photon of light is absorbed by one photosystem, which is paradoxically called photosystem 2 for historical reasons. As we have seen, that photon is used to produce a high-energy electron that is handed off to an electron transport chain. See Figure 1431. While traveling down the electron transport chain, the electron drives an H+ pump in the thylakoid membrane and creates a proton gradient in the manner described previously for oxidative phosphorylation. An ATP synthase in the thylakoid membrane then uses this proton gradient to drive the synthesis of ATP on the stromal side of the membrane. Figure 1432. In the meantime, the electron transport chain delivers the electron generated by photosystem 2 to the second photosystem in the pathway, called photosystem 1. There, the electron fills the positively charged hole that was left in the reaction center of photosystem 1 when it absorbed the second photon of light. Because photosystem 1 starts at a higher energy level than photosystem 2, it is able to boost electrons to the very high energy level needed to make NADPH from NADP+. See Figure 1432. The redox potentials of the components along this electron transport chain are shown in Figure 1433. In the overall process described thus far, we have seen that an electron removed from a chlorophyll molecule at the reaction center of photosystem 2 travels all the way through the electron transport chain in the thylakoid membrane until it winds up being donated to NADPH. This initial electron must be replaced to return the system to its unexcited state. The replacement electron comes from a low-energy electron donor, which in plants and many photosynthetic bacteria is water. See Figure 1431b. The reaction center of photosystem 2 includes a water-splitting enzyme that holds the oxygen atoms of 2 water molecules bound to a cluster of manganese atoms in the protein. Figure 1434. See also Figure 1432. This enzyme removes electrons one at a time from the water to fill the holes created by light in the chlorophyll molecules of the reaction center. When 4 electrons have been removed from 2 water molecules, which requires 4 photons of light, O2 is released. It is this critical process occurring over billions of years that has generated all of the O2 in the Earth's atmosphere. Chloroplasts can adjust their ATP production. In addition to carrying out the photosynthetic process outlined so far, chloroplasts can also generate ATP without making NADPH. To produce this extra ATP, chloroplasts can switch photosystem 1 into a cyclic mode so that it produces ATP instead of NADPH. In this process, called cyclic photophosphorylation, the high-energy electrons produced by light activation of photosystem 1 are transferred back to the cytochrome B6F complex rather than being passed on to NADP+. From the B6F complex, the electrons are handed back to photosystem 1 at low energy. Figure 1435. The net result, aside from the conversion of some light energy to heat, is that H+ is pumped across the thylakoid membrane by the B6F complex as electrons pass through it. This cycle increases the electrochemical proton gradient that drives the synthesis of ATP. Cells adjust the relative amounts of cyclic photophosphorylation, which involves only photosystem 1, and the standard non-cyclic form of photophosphorylation, which involves both photosystems 1 and 2, depending on their relative need for reducing power in the form of NADPH, and high-energy phosphate bonds in the form of ATP. Carbon fixation uses ATP and NADPH to convert CO2 into sugars. The light reactions of photosynthesis generate ATP and NADPH in the chloroplast stroma, but the inner membrane of the chloroplast is impermeable to both of these compounds, which means that they cannot be exported directly to the cytosol. To provide reducing power and energy for the rest of the cell, ATP and NADPH are instead used within the chloroplast stroma to produce sugars that can then be directly exported. This sugar production, which occurs during the dark reactions of photosynthesis, is called carbon fixation. The central reaction of photosynthetic carbon fixation, in which an atom of inorganic carbon, as CO2, is converted to organic carbon, is illustrated in Figure 1436. CO2 from the atmosphere combines with the 5-carbon sugar derivative, ribulose 1,5-bisphosphate, plus water to give 2 molecules of the 3-carbon compound, 3-phosphoglycerate. This carbon-fixing reaction, which was discovered in 1948, is catalyzed in the chloroplast stroma by a large enzyme called ribulose bisphosphate carboxylase, also called ribulose bisphosphate carboxylase / oxygenase or Rubisco. Because this enzyme works extremely sluggishly compared with most other enzymes, processing about 3 molecules of substrate per second, compared with 1,000 molecules per second for a typical enzyme, very large numbers of enzyme molecules are needed by the plant. Ribulose bisphosphate carboxylase often represents more than 50% of the total chloroplast protein, and it is widely claimed to be the most abundant protein on Earth. When carbohydrates are broken down and oxidized to CO2 and H2O by cells, a large amount of free energy is released. Clearly, the reverse overall reaction in which CO2 and H2O combine to make carbohydrate during photosynthesis must be energetically very unfavorable. For this process to occur, it must be coupled to an energetically favorable reaction that drives it. The reaction in which CO2 is fixed by Rubisco is in fact energetically favorable, but only because it receives a continuous supply of the energy-rich compound ribulose 1,5-bisphosphate, to which each molecule of CO2 is added. See Figure 1436. The energy and reducing power required for the elaborate metabolic pathway by which this compound is regenerated are provided by the ATP and NADPH produced by the photosynthetic light reactions. The series of reactions that allows cells to incorporate CO2 into sugars forms a cycle that begins and ends with ribulose 1,5-bisphosphate. Figure 1437. For every 3 molecules of CO2 that enter the cycle, 1 new molecule of glyceraldehyde 3-phosphate is produced. The 3-carbon sugar that is the net product and 3 molecules of ATP and 2 molecules of NADPH are consumed. Glyceraldehyde 3-phosphate then provides the starting material for the synthesis of many other sugars and organic molecules. The carbon fixation cycle, or Calvin cycle, was worked out in the 1940s and 1950s in one of the first successful applications of radioisotopes as tracers in biochemistry. Sugars generated by carbon fixation can be stored as starch or consumed to produce ATP. The glyceraldehyde 3-phosphate generated by carbon fixation in the chloroplast can be used in a number of different ways depending on the needs of the plant. During periods of excess photosynthetic activity, glyceraldehyde 3-phosphate is retained in the chloroplast, where it is mainly converted to starch in the stroma. Figure 1438. Like glycogen in animal cells, starch is a large polymer of glucose that serves as a carbohydrate reserve. Starch is stored as large grains in the chloroplast stroma. See Figure 1425b. And at night it is broken down to sugars to help support the metabolic needs of the plant. Starch forms an important part of the diet of all animals that eat plants. But the chloroplast is not merely a storage depot. Most of the glyceraldehyde 3-phosphate produced in chloroplasts is moved out of the chloroplast into the cytosol. Some of it enters the glycolytic pathway, see Figure 13.5, where it is converted to pyruvate. This pyruvate then enters the citric acid cycle in the plant cell mitochondria and leads to the production of ATP by oxidative phosphorylation. See Figure 1438. This is the ATP the plant uses for its general metabolism, and it is synthesized in mitochondria in the same way as in animal cells and other non-photosynthetic organisms. The glyceraldehyde 3-phosphate exported from chloroplasts can also be converted into many other metabolites, including the disaccharide sucrose. Sucrose is the major form in which sugar is transported between plant cells, just as glucose is transported in the blood of animals. Sucrose is exported from the leaves via the vascular bundle to provide carbohydrate to the rest of the plant. The origins of chloroplasts and mitochondria. It is now widely accepted that chloroplasts and mitochondria most likely evolved from bacteria that were engulfed by ancestral eukaryotic cells more than 1 billion years ago. See Figures 119 and 121. As a relic of this evolutionary past, both types of organelles contain their own genomes, as well as their own biosynthetic machinery for making RNA and organelle proteins. The way in which mitochondria and chloroplasts reproduce, through the growth and division of pre-existing organelles, provides additional evidence of their bacterial ancestry. Figure 1439. The growth and proliferation of mitochondria and chloroplasts is complicated by the fact that their component proteins are encoded by 2 separate genetic systems, one in the organelle and one in the cell nucleus. In the mitochondrion, most of the original bacterial genes have become transposed to the cell nucleus, leaving only relatively few genes inside the organelle itself. Animal mitochondria, in fact, contain a uniquely simple genetic system. The human mitochondrial genome, for example, contains only 16,569 nucleotide pairs of DNA encoding 37 genes. The vast majority of mitochondrial proteins, including those needed to make the mitochondria's RNA polymerase and ribosomal proteins, and all of the enzymes of its citric acid cycle, are instead produced from nuclear genes, and these proteins must therefore be imported into the mitochondria from the cytosol where they are made, discussed in Chapter 15. Like the mitochondrion, the chloroplast contains many of its own genes, as well as a complete transcription and translation system for producing proteins from these genes. Chloroplast genomes are considerably larger than mitochondrial genomes. In higher plants, for example, the chloroplast genome contains about 120 genes in 120,000 nucleotide pairs. These genes are strikingly similar to those of cyanobacteria, the photosynthetic bacteria from which chloroplasts are thought to have been derived. Even so, many chloroplast proteins are now encoded by nuclear genes and must be imported from the cytosol. The same techniques that have allowed us to analyze the genomes of mitochondria and chloroplasts have also permitted us to identify and explore the molecular biology of many microorganisms on the Earth. Some of these organisms thrive in the most inhospitable habitats on the planet. These include sulfurous hot springs or hydrothermal vents deep on the ocean floor. In these seemingly odd modern microbes, we can readily find clues to life's history, in the form of many molecules from which they are made. Like the fingerprints left at the scene of a crime, these molecules provide powerful evidence that allows us to trace the history of ancient events, permitting speculations on the origin of the ATP-generating systems that are found in today's mitochondria and chloroplasts. We therefore end this chapter with a discussion of the evolution of the energy harvesting systems that we have discussed in detail previously in this chapter. Oxidative phosphorylation might have given ancient bacteria an evolutionary advantage. As we mentioned earlier, the first living cells on Earth, both prokaryotes and primitive eukaryotes, may have consumed geochemically produced organic molecules and generated ATP by fermentation. Because oxygen was not yet present in the atmosphere, such anaerobic fermentation reactions would have dumped organic acids such as lactic or formic acids, for example, into the environment. See Figure 13.4a. Such an excretion of organic acids would have lowered the pH of the environment, favoring the survival of cells that evolved transmembrane proteins that could pump H+ out of the cytosol, keeping the cell from becoming too acidified. Stage 1 in Figure 1440. One of these pumps may have used the energy available from ATP hydrolysis to eject H+ from the cell. Such a protein pump could have been the ancestor of the present-day ATP synthase. As the Earth's supply of geochemically produced nutrients began to dwindle, organisms that could find a way to pump H+ without consuming ATP would have been at an advantage. They could save the small amounts of ATP they derived from the fermentation of increasingly scarce foodstuffs to fuel other important activities. Selective pressures, such as the scarcity of nutrients, might therefore have led to the evolution of electron transport proteins. These proteins allowed cells to use the movement of electrons between molecules of different redox potentials as an energy source for transporting H+ across the plasma membrane. Stage 2 in Figure 1440. Some of these cells might have used the non-fermentable organic acids that neighboring cells had excreted as waste to produce the electrons needed to feed the system. Some present-day bacteria grow on formic acid, for example, using the small amount of redox energy derived from the transfer of electrons from formic acid to fumarate to pump H+. Eventually, some bacteria would have developed H+ pumping electron transport systems that were so efficient that they could harvest more redox energy than they needed to maintain their internal pH. These cells most likely generated large electrochemical proton gradients, which they could then use to produce ATP. Protons could leak back into the cell through the ATP-driven H+ pumps, essentially running them in reverse so that they synthesized ATP, stage 3 in figure 1440. Because such cells required much less of the dwindling supply of fermentable nutrients, they would have proliferated at the expense of their neighbors. Photosynthetic bacteria made even fewer demands on their environment. The major evolutionary breakthrough in energy metabolism, however, was almost certainly the formation of photochemical reaction centers that could use the energy of sunlight to produce molecules such as NADH. It is thought that this development occurred early in the process of evolution, more than 3 billion years ago, in the ancestors of the green sulfur bacteria. Present-day green sulfur bacteria use light energy to transfer hydrogen atoms as an electron plus a proton from H2S to NADPH, thereby creating the strong reducing power required for carbon fixation. Figure 1441. The next step, which is thought to have occurred with the rise of cyanobacteria, see figure 1424, was the evolution of organisms capable of using water as the electron source for photosynthesis. This entailed the evolution of a water-splitting enzyme and the addition of a second photosystem, acting in tandem with the first, to bridge the enormous gap in redox potential between H2O and NADPH. See figure 1433. The biological consequences of this evolutionary step were far-reaching. For the first time, there were organisms that made only very minimal chemical demands on their environment. These cells could spread and evolve in ways denied to the earlier photosynthetic bacteria, which needed H2S or organic acids as a source of electrons. Consequently, large amounts of biologically synthesized fermentable organic materials accumulated. Moreover, O2 entered the atmosphere in large amounts. Figure 1442. The availability of O2 made possible the development of bacteria that relied on aerobic metabolism to make their ATP. As explained previously, these organisms could harness the large amount of energy released by breaking down carbohydrates and other reduced organic molecules all the way to CO2 and H2O. As organic materials accumulated as a byproduct of photosynthesis, some photosynthetic bacteria, including the ancestors of E. coli, lost their ability to survive on light energy alone and came to rely entirely on cellular respiration. Mitochondria probably arose when a primitive eukaryotic cell engulfed such a respiration-dependent bacterium, and plants arose somewhat later, when a descendant of this early aerobic eukaryote captured a photosynthetic bacterium, which became the precursor of the chloroplast. Once eukaryotes had acquired the bacterial symbionts and became mitochondria and chloroplasts, they could then embark on the amazing pathway of evolution that eventually led to complex multicellular organisms. The lifestyle of Methanococcus suggests that chemiosmotic coupling is an ancient process. The conditions today that most resemble those under which cells are thought to have lived 3.5 to 3.8 billion years ago may be those near deep ocean hydrothermal vents. These vents represent places where the Earth's molten mantle is breaking through the crust, expanding the width of the ocean floor. Indeed, the modern organisms that appear to be the most closely related to the hypothetical cells from which all life evolved live at high temperatures, 75 degrees Celsius to 95 degrees Celsius, close to the temperature of boiling water. This ability to thrive at such extreme temperatures suggests that life's common ancestor, the cell that gave rise to bacteria, archaea, and eukaryotes, lived under very hot anaerobic conditions. One of the archaea that live in this environment today is Methanococcus janisaki. Originally isolated from a hydrothermal vent more than a mile beneath the ocean surface, the organism grows entirely on inorganic nutrients in the complete absence of light and gaseous oxygen, utilizing as nutrients hydrogen gas, H2, CO2, and nitrogen gas, N2, that bubble up from the vent. Its mode of existence gives us a hint of how early cells might have used electron transport to derive their energy and their carbon molecules from inorganic materials that were freely available on the hot early Earth. Methanococcus relies on N2 gas as its source of nitrogen for organic molecules such as amino acids. The organism reduces N2 to ammonia, NH3, by the addition of hydrogen, a process called nitrogen fixation. Nitrogen fixation requires a large amount of energy, as does the carbon fixation process that the bacterium needs to convert CO2 into sugars. Much of the energy required for both processes is derived from the transfer of electrons from H2 to CO2, with the release of large amounts of methane, CH4, as a waste product, thus producing natural gas and giving the organism its name. Figure 1443. Part of this electron transfer occurs in the membrane and results in the pumping of protons, H+, across it. The resulting electrochemical proton gradient drives an ATP synthase in the same membrane to make ATP. The fact that such chemiosmotic coupling exists in an organism as primitive as Methanococcus suggests that the storage of energy derived from electron transport in an H+ gradient is an extremely ancient process. Thus, chemiosmotic coupling is likely to have fueled the evolution of nearly all life forms on Earth. Essential concepts. Mitochondria, chloroplasts, and many bacteria produce ATP by a membrane-based mechanism known as chemiosmotic coupling. Mitochondria produce most of an animal cell's ATP, using energy derived from oxidation of sugars and fatty acids. Mitochondria have an inner and an outer membrane. The inner membrane encloses the mitochondrial matrix, a compartment that contains many enzymes, including those of the citric acid cycle. These enzymes produce large amounts of NADH and FADH2 from the oxidation of acetyl-CoA. In the inner mitochondrial membrane, high-energy electrons donated by NADH and FADH2 pass along an electron transport chain, the respiratory chain, eventually combining with molecular oxygen, O2, in an energetically favorable reaction. Much of the energy released by electron transfers along the respiratory chain is harnessed to pump H+ out of the matrix, thereby creating a transmembrane proton H+ gradient. The proton pumping is carried out by three large respiratory enzyme complexes embedded in the membrane. The resulting electrochemical proton gradient across the inner mitochondrial membrane is harnessed to make ATP when H+ ions flow back into the matrix through ATP synthase, an enzyme located in the inner mitochondrial membrane. The proton gradient also drives the active transport of metabolites into and out of the mitochondrion. In photosynthesis in chloroplasts and photosynthetic bacteria, high-energy electrons are generated when sunlight is absorbed by chlorophyll. This energy is captured by protein complexes known as photosystems, which in plant cells are located in the thylakoid membranes of chloroplasts. Electron transport chains associated with photosystems transfer electrons from water to NADP+ to form NADPH. O2 is generated as a byproduct. The electron transport chains in chloroplasts also generate a proton gradient across the thylakoid membrane. As in mitochondria, this electrochemical proton gradient is used by an ATP synthase embedded in the membrane to generate ATP. The ATP and the NADPH made by photosynthesis are used within the chloroplast to drive the carbon fixation cycle in the chloroplast stroma, thereby producing carbohydrate from CO2. Carbohydrate is exported to the cell cytosol, where it is metabolized to provide organic carbon, ATP, mostly via mitochondria, and reducing power for the rest of the cell. Both mitochondria and chloroplasts are thought to have evolved from bacteria that were endocytosed by primitive eukaryotic cells. Each retains its own genome and divides by processes that resemble a bacterial cell division. Chemiosmotic coupling mechanisms are widespread and of ancient origin. Modern microorganisms that live in environments similar to those thought to have been present on the early Earth also use chemiosmotic coupling to produce ATP.